a pleasure to be here, and I am very excited to be here to talk about Genetics 101 and feed efficiency with you. So briefly tonight, I'll talk a bit about myself, some of the pressures we're seeing for more sustainable beef, as well as Genetics 101 and feed efficiency. So I see that quite a few of you are phenotyping and genotyping your animals, so this might be a refresher for you. The power of genomics, how it can bring our industry to the forefront of agriculture, and a little bit about what our research group is working on. So firstly, for those who don't know me, a little bit about myself and my background. I was born and raised in Hamilton, Ontario, and Hamilton is a big city, and you might be wondering how I found myself in agriculture. My parents always told me that Canada is full of opportunities, and I was fortunate to find those opportunities in agriculture, especially in beef. So I found a lot of my interests in beef research and did my PhD in beef cattle feed efficiency and genetics. I then worked at Agriculture Canada and Genome Alberta during COVID, and I decided to come back to U of G as a researcher. So thank you so much for having me tonight. And so I mentioned that there's a pressure for more sustainable agriculture and more sustainable beef production. And we are seeing this at a global, federal, and provincial level. Just as an example, I listed here some of these initiatives. We have the Global Methane Pledge, which is a worldly initiative to reduce emissions by 30% by 2030. At the federal level, we saw Genome Canada invest 30 million into a climate action initiative for research to mitigate emissions. We have the Canadian beef industry goal to reduce emissions by 33% by 2030. And these are just some examples of how these pressures often direct our research or also help direct our decision-making on farm and planning our research. But it's just very clear that one of the main goals is to have more environmentally and economically sustainable agriculture. And as many of you know, one of these approaches is to improve genetic selection for feed efficiency for more sustainable production. For those that aren't familiar, feed efficiency is a measure of the ability of an animal to convert its feed into a product like meat or milk. So for a more efficient animal, that animal can produce more product while consuming the same amount of feed compared to another animal. And as feed costs are the most highest and variable cost in production, and feed efficiency is related to methane production, there is potential for improved feed efficiency to have economic and environmental benefits. And from our research, we understand that feed efficiency is heritable. So this is a characteristic that can be passed down from parent to offspring. That means we can genetically select or breed for improved feed efficiency. But you might be wondering, what is the economic impact of producing more sustainable beef? And is it even possible to be sustainable and profitable and produce good beef at the same time? And the answer is yes. So we have more feed efficient animals, which would lead to less resources needed to make the same amount of product. And studies have shown a favorable phenotypic relationship between feed efficiency and methane production. And now we're currently studying the genetic relationship between feed efficiency and methane production. And from research, we understand that methane is a loss of energy that the animal could be putting towards more gains or lean yield. And also a former study showed that breeding for lower methane and higher efficiency can reduce feed costs by up to $108 per animal per year. And this leads me to the potential of using genetic and genomic selection to improve feed efficiency. So with genetic or genomic selection, it all starts with collecting a phenotype and a genotype. And firstly, what is a phenotype? A phenotype, also known as a trait or a phenotypic record, is simply the observed trait or characteristic of an animal. So just as an example, this could be a hair color, feed intake amount, body weight at a certain age or production phase, marbling or milk yield. And these phenotypes in industry are often economically important. So if we want to produce more milk or more meat, then we'll have to measure that. So shown here is just a summary table of some of the traits or phenotypes that we collect in our research experiments. And I mentioned that we need both a phenotype and a genotype. So now, what is a genotype? And I apologize if this is a refresher for many of you. A genotype is the genetic information of an animal or DNA information. So you may hear that we genotype or we DNA test our animals to get genetic data to find mutations in their DNA. And these mutations in their DNA could be impacting their phenotype. So if they're more efficient or less efficient, or if they produce more methane or less methane. So this can be done at any point in their life to DNA test them because genetics are the same throughout an animal's lifetime. And you may find more benefit in DNA testing at birth so that you can make management decisions, for example, detecting genetic defects. You can verify their parentage. You can determine if that animal is genetically valuable and maybe could be used for breeding. And you might be wondering, how do you genotype an animal or collect the DNA from an animal? So on the side here is just an example of how that works for one of the breed associations that I went to their AGM to talk about their process. This is an example, but to save time, if you want to learn more about the process, you can contact your breed association or services like egg sites to get more details. But it is a straightforward process. It can be as simple as collecting a hair sample and sending that sample to a service like Neogen, which there's generally a process already in line for you to do that. And then you would get your genetic prediction values back to you. I'll talk more about that later. But I do want to mention that improving an animal through genetics is permanent and cumulative, which means that when we use breeding strategies, the genetic gain at each generation is permanent and continues to improve. So if you're breeding for more marbling, you would expect that marbling will increase at each generation. So next we'll talk about the approaches to make selection decisions for traits, and that includes genetic evaluation and genomic evaluation. And I'll tell you the differences. So firstly, genetic selection or genetic evaluation uses EPDs or expected progeny differences. If you are familiar with EPDs, feel free to shoot in the chat like, yes, I know about EPDs, or maybe, oh, I'd love to know more about this. I'm going to ask BCRC for some information or my breed association. EPDs are also known as a breeding value, and it's a prediction of the ability of the parent to transmit its genetics to its offspring. So this is a calculated value using a mathematical model that will tell you how genetically valuable that animal is. This calculation requires a phenotype and pedigree data. So performance data or phenotype, example, body weight, feed intake, marbling, or as well as pedigree data, so the family history. When we have the phenotypic data and the pedigree data, we can provide weekly breeding value or EPD predictions of your animals and other animals. You can actually rank these animals, your animals, based on their breeding values for different traits, as well as if you have multiple breeding objectives, different selection indexes can be implemented for that. You can then select sires and dams with the highest EPD for breeding, for example. This is a very effective approach to make breeding decisions, and it is low cost. You can achieve genetic gain on easy to measure traits like marbling, feed to gain, weaning traits. But if we're comparing genetic evaluations with genomic evaluations, there can be some limitations. It is a very effective approach, but there may be lower accuracy to predictions. For example, if we have errors in pedigree data, or the pedigree relationships aren't as accurate as the genotypic data. We may have lower selection efficiency on difficult to measure traits like feed efficiency. And we might see a slower rate of genetic progress compared to genomic selection, but it is still very effective. Now, moving forward to genomic selection, we are entering the world of EPDs with the added information of genetics. So we call this genetically enhanced EPDs, or GE-EPDs. And feel free to take pictures of the slides, and I'll share this as well after tonight's webinar. This is when we add genotypic data or DNA test results to enhance the predictability of our EPD predictions. So it increases the accuracy. And just to highlight, it's the same as EPDs, but we add genetic data to that calculation. Some of these benefits will include that the EPD values adjust with added genomic added genomic information. So for example, if we test an animal that has a strong genetic relationship with another animal that is already proven to excel in marbling, then that newly tested animal's marbling EPD will increase. So it helps all of the calculations when we add more genetic data, even for one animal. Some of the limitations we observe is that we require a large training population. So we need a population of animals with both genotype and phenotype to make these calculations, which is why it's so important to have both. The traits of interest may be costly and laborious or time-consuming to measure, for example, feed efficiency. And genotyping costs can be high, but we have seen a decrease in costs over time significantly as technology improves. And then this is just showing an example of the power of genomic selection. So here is showing the impact of genomic selection in Canadian dairy cattle, where they're making selection decisions based on genomic information, doing that DNA testing and inputting that phenotypic data, especially for milk yield. This doubled the rate of genetic gain and increased total milk yield with less animals. And we can see the exponential increase since 2009 when genomic selection was implemented. Now, this happened for multiple reasons. They have a large reference population with over 85% of submissions being from one breed, and that's Holstein. And they frequently do genetic testing of offspring. Here we can see another example comparing the impact of genomics before and after, so of key traits. So the units before using genomics in dark blue and the units after using genomics in light blue on these key traits like milk yield, fat and protein percent, longevity and fertility. And we can see almost a doubling of improvements after using genomics. So we have observed many great things resulting from genomics. And just summarize here the accurate parentage verification and discovery, which is more reliable than pedigree data. We have higher accuracy of selection for younger animals. Faster rates of genetic progress became a reality, and also improved new and novel traits as we have improving technology to measure traits indirectly, like feed efficiency. I'm sure Kelly, our next speaker, will likely touch on those emerging tools. And then what is the future of genomics for the industry? We expect a continued increase in genotyping and phenotyping, as well as the use of commercial genotyping services from your breed association or services like egg sites. A continued use at the seed stock breeder level, so lots of benefits to cow, calf, and feedlot producers that are seen down the value chain. increasingly lower cost for genotyping. And with all of that together, it would result in a growing reference population with both types of information, genotypes and phenotypes. So we're also seeing new traits and technology to help producers. And this new technology would allow for us to measure very costly and difficult to measure traits like feed efficiency. So it would still be highly accurate, more efficient, and lower cost to measure data that will greatly improve genomic selection. But the key message is that the future to genomics will greatly depend on us working together to record phenotypes and collect genotypic information. So this talk was just a brief overview of genomics, but if you got excited about genomics and genetics and would like to start taking your first steps or even just learn about what services are available to you, for example, even talking about how to define breeding objectives or maybe just ask a few more questions about it, I would highly recommend speaking to any of these individuals. Erin is a manager of research and innovation at Egg Sites, and they offer ultrasound measurements and conduct the genomic evaluation calculations. As well, BCRC website has so many fact sheets and summaries about this information. What is an EPD? What are selection indexes? What traits are being measured? Ellen Crane is a great resource for that. And of course, you can connect with our research group anytime. I work with Dr. Angela Canovis, and we can give you many resources and answer as many questions as we can. As well, your breed association website may even offer these resources and services for you already and guide you on that process. And very briefly, to talk a bit about the work we do in the Canovis lab, we study the genetics of these desirable traits, feed efficiency, methane production, to improve breeding strategies. So our main goal is to help beef systems become more environmentally sustainable, but still profitable and produce good quality beef. We work with herds at the Ontario Beef Research Centre, the newly updated research facility. And then very briefly, these are some of the phenotypes we collect, and of course, genotyping the animals as well. And this is key for our research. Many of these measurements are actually being collected as an ongoing basis at the center. And that information will be used to calculate feed efficiency and study the genetics underlying these traits. With that, I'm going to end with some acknowledgements, and I'd love to thank all of these industry partners and associations that have supported us for many years. We really can't do this without them. And Angela made this beautiful slide with all of our students to thank you from us researchers. We thank you and have huge gratitude to work with you. And just a huge thank you from the Canovis Lab.